ओके। सो जीनोम एक क्या है? डीएनए के जितने मटेरियल है वो से जीनोम कहते हैं। जीनोम बहुत सारी चीज़ें होंगी जैसे यूकैरियोट, प्रोकैरियोट एंड सम वायरस। नाउ जीनोमिक डीएनए, जीनोम्स आर एक्चुअली मेड अप ऑफ डीएनए, बट कुछ वायरस होते हैं उनमें आरएनए के जीनोम्स होते हैं। So the eukaryotic genomes consist of two parts, nuclear genome and organelle genome. So nuclear is nucleus and organelle में mitochondria and chloroplast रहेगा. The amount of DNA present in haploid genome species is called C value. Wow. The amount of DNA present in haploid genome is called C value. C value is the characteristic of each species. The value range from 10 to the power 6 base pair, as in smallest prokaryote mycoplasma, to more than 10 to the power 11 base pair for amphibians. The genomes of high organisms contain large excess of DNA. Okay, so 10 to the power 6 हो तो वायरस में सबसे कम है, माइकोप्लाज्मा एल्गे में भी कम है. Okay. To the figure point 1.1 is showing the haploid genome ranges of phyla. So DNA content of the haploid genome is related to morphological complexity of low eukaryotes, but it varies extensively among the high eukaryotes. In low eukaryotic organisms like yeast, amount of DNA increases with increasing complexity of organism. However, high eukaryotic there is no correlation between increased genome size and complexity. This lack of correlation between genome size and genetic complexity is referred to C value. For example, man is more complex than amphibians in terms of genetic development, but some amphibians cells contain 30 times more DNA than human cells. Moreover, the genome of different species of amphibians can vary hundredfold in their DNA content. Saccharomyces has 12 genome, Thaliana which is mustard plant 120, while human Homo sapiens has 3300 and Barley has 2300. So genome complexity is total length of different sequence of DNA. What is genome complexity? Total length of different sequence of DNA. Genome complexity can be measured through renaturation kinetics of denatured DNA. Renaturation DNA can occur through complementary base pairing. Renaturation of DNA depends on random collision of contemporary strands and follows second order kinetics. A DNA associated a DNA association reaction is described by quad value, quad half. If large DNA is shared into uniform fragment and allowed to renature, then the rate of renaturation of denatured DNA is expressed as dC by dt. Okay, that is minus Kc square, where K is second order rate constant, C is concentration of single stranded DNA at time t, and second order rate equation of for two complementary strands coming together is given by the rate of decreased in C. Starting with concentration C naught of completely denatured DNA at T equals 0. भाई ये क्या है क्वार्ट वैल्यू एंड ऑल वैल्यू फक. Okay, so repetitive linear sequences were first identified as studies rate with DNA reassociation. What relative rates of reassociation are expected for sequence repeated 1000 times? The genes compared to genes with only single copy. DNA reassociation is a biomolecular reaction, so the reaction rate is proportional to the square of the DNA concentration. A sequence repeated 1000 times in the genome that would definitely be associated with 10 to power 6 fold.
Hello.